Okay, so welcome to this Friday talk. Ramon couldn't make it today, so I'm taking the lead. It's my pleasure, actually, to introduce to you Elisabeth Deschaseaux. From the name, you can easily guess she's French, even though she's also Australian. She's been 18 years now in Australia. So probably she's even more Australian than French, but who knows? I mean, it's up to her, of course. There's no conflict in that. Liz, I mean, the good thing of Liz is that she's now visiting us, but as I sent a message to you guys, she's visiting us to get some training on the analytical system that we have, and this is why we invited Charel to come again over and train Liz about the analytical system, but she's heading back to Australia soon, next week, and then she'll join the group, she'll join the department and the ICM after summer. So she'll be new staff next year for at least a couple of years. Who knows what life will have for her. So she did her bachelor in Bordeaux when she was in France, and yesterday or the day before she was telling me that she got a bit into neuroscience and things like that. She's a biologist, and finally she kind of changed her mind. As a neuroscientist, she decided to go more into environmental sciences and marine sciences, and then she moved to Canberra, to the University of Canberra, to do her honors in marine sciences, and then she moved to Lismore, where she did her PhD on corals, and this is what she will tell us about, with a few visits to the University of Technology, Sydney, where we have colleagues as well. So she's been kind of around Australia for a while. And then for the last few years, she's been working hard, can you imagine, in the Great Barrier Reef in Australia, doing beautiful research there on volatiles, and this is what attracted our attention. Of course, I've been following her for many years, and at some point I decided to chase her and say, "Hi, Liz, is it time by chance to change place and come to Barcelona? I would be pleased to have you in my group." And eventually she said yes. So I feel very privileged to have her joining us. So Liz, the floor is yours.
Thank you. Well, thank you, Raphael, for this great introduction, and hi, everyone. Should I say hola a tutem? Soy Liz. Un player estar acá. So this is the extent of my Catalan for now, but I swear to you I'm going to practice. So yeah, what I'm going to tell you today, I'm going to present some work that I've done on emissions of volatile organic compounds in coral reef ecosystems. So as part of my last postdoc at Southern Cross University, which was also part of the Reef Restoration and Adaptation Program, which I will tell you a bit more about. So yeah, well, I mean, Raphael already introduced me, so I'm going to skip really quickly on that. But yes, I did my undergrad in France, and then all the rest of my studies in Australia, and I did my PhD on dimethylated sulfur compounds in acropora coral. And then a few postdocs at UTS and mainly at Southern Cross University. And the last one was part of the Reef Restoration and Adaptation Program. So just a bit of background on biogenic volatile organic compounds for those of you who are not too familiar with them. They're essentially trace gases that are produced by both the marine and terrestrial environments. And they can have different effects on climates by either increasing global warming or decreasing global warming, so through direct or indirect effects. So mainly the direct effect on global warming is that BVOCs contribute to ozone formation, and they contribute to CO2 emissions as well. And also when they are emitted in the atmosphere, they can increase the lifetime of methane. So that's the indirect effect, which will lead to, they increase the lifetime of methane because they react with hydroxyl radicals in the atmosphere. And so therefore that will create indirectly a warming effect. And the two cooling effects are that BVOC contribute to the formation of secondary organic aerosols in the atmosphere that have scattering properties and therefore reflect light to space. So that's the direct effect. And indirectly, because they can contribute to, these aerosols can contribute to CCN, to cloud condensation nuclei, so which I will get into more details a bit later. But the main question, I guess, when we look at BVOCs is we want to understand whether these emissions, when they are placed in global models, whether these emissions are likely to enhance or inhibit global warming under future climate change conditions. So two of the BVOCs that I worked on are dimethyl sulfide and methanethiol, and these two BVOCs are sulfur compounds that play key roles in the global sulfur cycle. They originate from DMSP, or dimethyl sulfoniopropanate, that is produced by many types of phytoplankton, macroalgae, and also coral. So the reason why DMSP is present in high concentrations in coral reef ecosystems is that corals not only produce DMSP through the symbiotic microalgae that they live with, but also through the animal host itself. So that's why coral reefs are considered hotspots for DMSP. So the DMSP, then, there are two different pathways. DMSP can get cleaved into DMS, or dimethyl sulfide, which is associated with the smell of the sea, and is probably one of the most studied BVOCs in the marine environments. So this process occurs through different DMSP layers, so it's an enzymatic process. And then there is a process of demethylation that leads to methanethiol, mainly mediated by bacteria. And in fact, the demethylation process is the most dominant process by which DMSP is degraded in the marine environments. But often concentrations of DMS are 3 times greater in the water and in emission fluxes than methanethiol or MESH. So most likely because MESH is used by bacteria as a source of sulfur and carbon and is used to make methionine. And also because the lifetime of methanethiol in the atmosphere is often a lot less than the lifetime of DMS. So that's the two main difference in between these compounds. And the reason why DMS has been so extensively studied as a BVOC from marine systems is that it is known to, when it is emitted into the atmosphere, so a small portion of the DMS that is produced through cleavage and enzymatic activity gets released into the atmosphere by wind and wave actions, usually about 10% of it. And this DMS gets oxidized into methane sulfonic acids and sulfur dioxide that gets further oxidized into sulfuric acid and sulfate aerosols. And these compounds mixed with sea salt are actually the base, can either condensate into existing molecules, or they can create a cloud condensation nuclei that will therefore have an impact on the albedo of the clouds, meaning the cloud's capability or capacity to reflect light back to space in an efficient way. So there is a theory behind that, the CLO hypothesis, in fact, that I won't get too much into details today, where we think that there is a feedback effect of the environment on the DMS production. That part of DMS contributing to CCN we know, but the part of the environment potentially having a negative feedback effect on climate that we don't know, and it's still a theory today. Where MESH plays a role is that actually in recent years, so all of the attention has been given to DMS mainly, but recently, in recent years, MESH has been shown to contribute actually to 30% of the SO2 formation in the atmosphere. So we believe that this BVOC also has cooling properties that need to be explored, and so more and more attention is actually given to research on methanethiol, which is in fact part of the work, hopefully, that I will be doing with Raphael and his team here. Besides DMS and methanethiol that we just talked about, there are other compounds and other BVOCs that are considered climatically relevant, and the two that I targeted for the work that I did at SCU were isoprene and monoterpenes. So these two compounds are BVOCs that are actually really present on Earth. They are 50% for isoprene and 15% for monoterpenes. They contribute, to 50 and 15% of all the BVOCs that you can find on the planet. But they are mainly sourced from terrestrial ecosystems, although in recent years many efforts have been made to measure them in the marine environment, and they have been found actually in many cultures of phytoplanktons, and they contribute to climate regulation in a way through the formation of secondary organic aerosols, so in the same way as DMS and methanethiol, but also they contribute to ozone formation, so they are considered to have a cooling and warming effect. So these are the four compounds mainly that I concentrated on for this project. So just a little bit of background on so why BVOCs in coral reef ecosystems and why that's actually triggered our interest was that the Great Barrier Reef has been experiencing many mass coral bleaching events over the past decades, with 5 of them occurring in the past decade actually. So 2016, 17, 20, 22, and you can see on the map there that it first started in the north of the Great Barrier Reef because it's the warmer part of the Great Barrier Reef, but then that it started to spread to the rest of the GBR, and that it's becoming more severe, more frequent, and more widespread with time. And in fact, sadly, this year, so in Australia, we have just passed the summer. This is the most severe mass coral bleaching events that we've recorded for the Great Barrier Reef. They're estimating right now, of course, every summer, some researchers from the Australian Institute of Marine Science go and evaluate the health and the state of the reef, and right now they're estimating 70% of the Great Barrier Reef being bleached, which doesn't mean 70% dead, but it's massive. And it's actually this bleaching event is a worldwide event this year, so we have coral reefs from all around the world being bleached, sadly. So the incentive in this context, in fact, of mass coral bleaching events, the Reef Restoration and Adaptation Program has invested a lot of money to help the reef survive or adapt to climate change. In fact, it's more kind of giving time to the reef to adapt. It's buying time, even though we know that the solution is to reduce CO2 emissions, but at the rate we are going, and we need to come up with some band-aid solutions. So this is the world's largest effort to help a significant ecosystem like the Great Barrier Reef survive climate change. It's a multi-partner project that involves Southern Cross University, CSIRO, and AIMS, to just name a few. And the government invested basically $100 million to find solutions for the reef. And the program that I was part of, so it's a sub-program of RRAP, got given $10 million. So it's the cooling and shading sub-program of RRAP that I was part of. And you can see that there are many, including some that they are trying to select some coral genetically to repopulate the Great Barrier Reef with species that are more thermal tolerant, either through the animal host or through the symbionts. I'm saying this because we actually did some collaboration that I will show you a bit later. So the idea behind the cooling and shading sub-program of RRAP is basically to reduce the amount of solar radiations reaching the reef to prevent or reduce coral bleaching. So they tested two different technologies, which is marine cloud brightening and fogging. So marine cloud brightening is a more long-term solution they see, where you spray filtered seawater into existing clouds, and the idea is to change the microphysics of the clouds and make the clouds more, to increase the albedo of the clouds, so make them more reflective. And fogging is essentially more of a short-term solution, and it will be during a crisis like this of mass coral bleaching events, having like fog over the reef where you create like a reflective surface basically to reflect light back to space. So this is pure geoengineering, and this is what they basically tested for the cooling and shading sub-program. So where the BVOC measurements that I took, that I made actually were part of RRAP, the idea was to measure BVOCs under many different conditions of temperature, light, nutrients, salinity, chlorophyll, and so on, to, well, first have a background measurements of these BVOCs from coral reef ecosystems, and determine the main drivers of them in coral reef ecosystems, and then feed them into existing models to make accurate predictions on BVOC's emissions under future climate change scenarios, with and without the RRAP intervention of marine cloud brightening and fogging, and anticipate the impacts of these future BVOCs on climate, knowing the impact that, knowing the role that they play in climate regulation. So we used a proton transfer reaction mass spectrometer, which is the instrument that I will also be playing with in Raphael's lab. So the idea behind the PTRMS is that we use ultra-pure water to generate a primary ion that then reacts with compounds and protonates the BVOCs. So it's essentially the mass of the BVOC plus one that you are measuring. And it's considered a soft ionization method because it leads to very little fragmentation compared to other techniques. And you have two different mass spectrometers that are associated with those instruments, either a quadruple or time of flight. So I have highlighted the quadruple here because it's the instrument that I used for the first campaign that I ran. And also the cool thing about this instrument is that it only reacts with compounds that have a greater proton affinity than that of water. So it doesn't react with the main component of air like nitrogen or oxygen or methane, CO2. So it is specific to trace volatiles like the ones we are interested in. The first campaign that we conducted as part of RRAP was basically, so I was taking all of the background measurements with the PTRMS, and these guys were doing the marine cloud brightening and fogging on the site, so I was not involved in that. It was just part of this project. And so basically we had two voyages, essentially, in December 21 and 22, which in Australia is the summer month. And so, yeah, basically one on RV Guardian and RV Magnetic, and the second voyage that we did in February had two legs, one at the, yeah, one on both research vessels, and we were 100 kilometers north of Townsville, and we covered many different reefs here that are like indicated with a star on the map. I won't bore you with the names of the reef, but it's essentially, yes, inside the mid-shelf lagoon and about 100 kilometers from the coast of Townsville. The setup that we had on the boat, so that was the setup on the RV Guardian, is we were essentially alternating in between atmospheric sampling and dissolved sampling every hour. That was our atmospheric inlets on the starboard side of the ship. And we were using a shower head exchanger for measuring our gases in equilibrium. So we were pumping basically water continuously with a metal-free pump into the shower head exchanger, and we were measuring the BVOCs in the headspace of the shower head. So that's how we did our dissolved measurements. And you can see that's like a hand-built calibration unit that we used for calibrating our instruments on a daily basis. And that was our setup on the other ship, basically. So you can see the same, like the PTRMS here, calibration unit, the shower head exchanger. And we were also measuring wind speeds with a sonic anemometer to have the three-dimensional wind velocity. And we were able to correct for the movement of the platform by having an inertial measurement unit, both measuring at 100 Hertz, so we could correct the wind for the movement of the platform. And that was important for measuring our fluxes accurately, which in the case, well, we wanted to, I actually wanted to look at two different approaches to estimate the fluxes in that case, which we couldn't do with that particular instrument. I was interested in doing an eddy covariance approach, but that's something that I'm intending to do with another paper, because we didn't have quite the right time resolution that we need for eddy covariance. But essentially what we did with the bulk approach was to using our dissolved and atmospheric concentrations and working out the gas transfer velocity constants based on a numerical scheme by Johnson et al. That gives you basically for any gas, you can work out the water side and air side gas transfer velocity. So the fluxes that those are the flux estimates for the different compounds that we measured. So the ones I highlighted at the bottom, we can disregard them for the purpose of this presentation. It was just that I had a peak arrow on the side, and I was measuring CO2 and methane in the water column and in the atmosphere as well. But we can see DMS, methanesyol, isoprene, and monoterpenes in the two different voyages that we did. What essentially that showed us was that the reef was actually a net source of DMS, methane, and isoprene. To a certain extent, monoterpenes, but concentrations were well, emission fluxes were very low for monoterpenes in comparison. And also that it basically, there were the first flux measurements for the Great Barrier Reef in terms of methane, isoprene, and monoterpenes. And we could see, by running some multilinear regression analysis, we wanted to isolate the main drivers of these fluxes. And of course, unsurprising, so we basically run a multiple linear regression analysis on all the different parameters that we measured as part of this, alongside these fluxes, so which was wind speed, wind direction, relative humidity, air temperature, dew points, light, the different latitude and longitude of our voyage, sea surface temperature, salinity, and the corresponding dissolved and atmospheric concentrations. And of course, unsurprisingly, wind speed and dissolved concentrations were the main drivers of the fluxes because they are co-factors in the flux calculations. But also interestingly, for isoprene and monoterpenes, the corresponding atmospheric concentrations also played a role in the flux estimates, which we find was interesting and probably are linked to the chemical properties of those compounds being more or less soluble. And if we looked at, we also looked at those compounds on the topographic map of the region. So the greater the gradient, the higher the concentration. And so we could find a coastward gradient for most of these compounds, especially for monoterpenes. And we could see for DMS, MESH, and isoprene that there was also not only a coastward gradient, but also a gradient along the coastal strip, actually, that separates the mid-shelf lagoon and the coast. So that was surprising, not so much for compounds like isoprene or monoterpenes because they have a terrestrial source, and we were therefore expecting them to be closer to the coast. But it was quite surprising for compounds like DMS and MESH that we were expecting to see more like in higher concentrations within the coral reef ecosystems that we believe are more concentrated in DMSP and therefore will produce more of these BVOCs. So we think it's most likely due to the fact that the coastal strip that separates the lagoon and the coast is actually really enriched in nutrients. And when we looked at, well, in fact, chlorophyll A, which we don't have the data for December 2021, but only for February 2022, we can see that there is definitely a coastward gradient as well for chlorophyll. And we looked also at sea surface temperature that was greater, also closer to the coast. So we suspect that these two factors are or were influencing potentially our BVOC fluxes. Another interesting thing that we looked at is we plotted MESH and DMS, the ratio of the two, so in the atmosphere. So these again are for the two voyages separately. I forgot to say that the graphs that I showed you earlier were for the two voyages combined. But so here is the ratio for the atmospheric concentrations, the ratio for the seawater, and the ratio for the flux. On average, the ratio in the atmosphere was 1:1 for these two compounds, and in the water and for resulting emission fluxes, it was more around 25%, which actually is more what we see in the rest of the literature. So the 25% is usually what you will expect. You will expect MESH to correspond to a small portion of the DMS. In the atmosphere, what we believe is happening is probably an artifact of our analysis because, as I said to you earlier, the instrument that we used was a quadruple PTRMS, which means that we have a much lower mass resolution. And we looked at basically targeted peaks at the mass of interest. Meaning that now that we have the time of flight mass spectrometer, we could see some interfering peaks at the mass of methane thiol in the atmosphere. So we believe that this 1-to-1 ratio that you see here, which is in fact higher at times, is most likely an artifact of this other compound that might be present in the atmosphere. But not so much in the water phase. You only see one clear peak for methane thiol in the water phase. So that's a paper that was just recently submitted to JGR Ocean, and we are hoping to see what they think of this first paper. Another campaign that we conducted was in February 2023, so just last year, and it was one of the biggest campaigns as part of the Reef Restoration and Adaptation Program, because we had a spraying vessel that was doing the marine cloud brightening and fogging intervention trials, and we had a measuring vessel that was measuring directly inside the plume, and a team in Gladstone, kilometers away, that was measuring the composition of the clouds with an aircraft, a drone team on One Tree Island doing the drone work, and us on Heron Island doing all of the background atmospheric and dissolved measurements. So that was quite a huge deployment. So for us, this time, we used the PTRMS, but we at this time had access to the time of flight mass spectrometer that is very similar to the one that you have at the ICM. And so we had, that was our beautiful air conditioning system that we brought with us, because we were in a space that had no air conditioning, and the PTRMS is not happy, or I should say the turbo pumps of the PTRMS are not happy at temperatures higher than 30 degrees. So we brought that with us, and so that's the instruments. We also had the Picaro on board to measure CO2 and methane in the same way as we did for the first campaign in 21 and 22. And we had our water inlets pumping water continuously into the shower head exchanger. That's me being very proud of my pump system after days of trying to fiddle with it, because it was a challenge to get to pump water from that far away from the reef. Same story, we had the sonic anemometer to measure the three-dimensional wind speed, and this time I really want to look at these flux estimates from two different approaches. So use a bulk approach, the same as what we did last time, but also explore the eddy covariance approach, because now we have an instrument that has a much higher time resolution. So in theory, with a high resolution wind speed and high resolution atmospheric concentrations, we should be able to also estimate fluxes with an eddy covariance approach and compare the two. So that's another paper that we are hoping to submit soon, that I'm still working on. So that's why I'm not presenting anything on it right now. But that's part of the work that we conducted last year. Another thing about BVOCs is that, so this is just the case of DMS and other related compounds, is that BVOCs in general can be involved in stress response in many marine and terrestrial organisms, in fact. And so in the case of DMS, it is known to be used as a thermoregulator, especially as a cryoprotectant in certain types of algae. An osmoregulator, it can also be a chemo repellent or attractant, so a signaling molecule for many different organisms. Well, in fact, it's like just a fun fact is that it's a chemical cue for the hatchlings of loggerhead turtles. That's the way they find their way to the ocean when they hatch. And they can, these compounds be also used as antioxidants in phytoplankton and coral, in fact. as they react with reactive oxygen species under oxidative stress. And so the idea was with a PhD student that I work with, Jasmine Haskell, the idea was to then, as part of the Arab cooling and shading, look at BVOC emissions in coral under stress. So we basically had different treatments of non-shaded with ambient temperature, and then temperature stress, mimicking bleaching, with and without shading conditions. So we were trying to mimic what will happen if the strategy of the cooling and shading were to work, and that's if we were able to apply a 30% shade on the reef, what will happen to the BVOC signature of our coral. So what we had, essentially, we had chambers that we connected in a closed loop system with the instruments, and we had a switchboard that we built together to actually alternate in between our chambers at the time. So what we had is we had a semi-continuous sampling of the chambers, and we could see live what was happening over time under all of our different treatments, alternating in between treatments. And that's the beautiful view from underneath with all the cables. It's controlled by an Arduino that controls the switching. And yeah, so basically that's what Jasmine was doing. We did an experiment on Heron Island and also at the measurement center in Coffs Harbour. And you can see here, I really like this photo actually of the coral, and you can see some of the bubbles, so some of the BVOCs, hopefully being emitted happily by the corals. So these are very preliminary results that Jasmine got for her PhD. We just looked at DMS, and you can see the different treatments of control, heat and shade, heat with no shade. And we could essentially see a greater signature with emission of DMS in the treated chambers, and we had a very similar response for MESH. One thing that is not shown in these graphs is that there was actually a very interesting shift in between those two compounds under stress. So what we had is often like first a peak of DMS that was released indicating stress, that when we visually saw that the coral were experiencing stress. But when we reached a kind of a non-return point where the coral was in fact dying, and we had to isolate some of in that particular experiment, some of the chambers because what we were measuring was just too high and saturating the system, was that MESH took over when the coral was dying. So we also took some samples from the inside of the chambers to look at the microbial composition, which we are still in the process of analyzing. But it will be super interesting to see and to link it with the microbial community if this switch in between DMS and MESH is actually potentially linked to a shift in the microbial composition. And the last experiment that we did was looking at coral stress, but in coral juveniles. So that's something that has never been done before, so we were really excited about it. We essentially had coral juveniles on those tiles. We had different treatments. This time we didn't look at shade, but mainly at temperature, because we wanted to have a good enough replication level for looking at two different types of juveniles. So the idea behind was that we collaborated with other members of ARAP who did work with selected coral. So either they did that through selective breeding. So they looked at parents that were performing the best under rapid heat stress, and they were breeding these colonies to, so the ones that were the best performers were called the winners, and the ones that performed the least were called the losers. And those respective parent colonies were bred together to have offsprings that were either considered heat tolerant or heat sensitive. So it was more looking at the heat tolerance, but through a coral point of view. And through the selective inoculation, it was a certain species of coral that had aposymbiotic inoculate, so without the symbionts, and they were inoculated with either heat-tolerant symbiodinium or heat-sensitive symbiodinium. All I can show you for now of this experiment, because we just conducted it now in February, was the fact that we had a very clear coral signature, and we were really happy about that because we went to the Australian Institute of Marine Science at CSIM, which has like an amazing sea simulator, and we didn't know whether we could even detect BVOCs from those really tiny organisms. And what we can see here is the trace of DMS only. The color coding is basically we had chambers that contain seawater blanks only, some that contains seawater blanks with blanks that were that only had the tiles, and because we wanted to eliminate the possibility that biofouling could contribute to DMS or other BVOCs. And we had tiles that had a certain number of coral juveniles on them. And we were so happy to see that the seawater blanks and the chambers that had blank tiles without any coral juveniles showed like a low signal, a background signal basically, and the ones that had coral showed a peak of DMS. So that was the first validation that we did before running our experiments. And Jasmine is in the process of analyzing all the data from this experiment. But at least we validated the fact that we could detect BVOCs from coral juveniles that had just been inoculated with symbionts. So everything will hopefully be in Jasmine's thesis, which we are hoping to, that she's still working on. So on that note, I just would like to acknowledge my supervisors for this work, Brad Eyre and Dan Harrison, who I did the work with ARAP. A really dear collaborator of mine, Erin Dunn, who became a mentor and has really helped me throughout this really cool process. The PhD student who I worked with, Jasmine Haskell here, and of course our funding bodies of the Great Barrier Reef Foundation and Australian Reef Crust. And yes, and Eric Whitstone, who helped us build the switchboard that we used for the chamber work that we did with Jasmine. So yeah, on that note, I'll be happy to answer any questions. I can hear you.
Hola? Ara? Thank you for the talk. It was very interesting. I have a question about how you mentioned you calculate fluxes using two approaches. One is the bulk approach, the other is the eddy covariance. Mm-hmm. Which variables did you measure for the eddy covariance? I saw the wind. Yeah. But were you able to measure any of the volatiles at the same precision that winds?
Yeah, so that's so we were still alternating in between. I haven't done the eddy covariance work yet, but that's definitely the approach I want to use. We were using actually the PTRMS then, which is the time of flight, and so we have a high time resolution, so we can measure at 10 hertz. So those ones, the wind speed and, just the wind speed was measured at 100 hertz. And we can decide on the time resolution of the PTRMS that we used. So the answer is yes, in fact, we can have a similar time resolution or narrow down the time resolution of our wind speed to match with the time resolution of our time of flight. And for eddy covariance, the minimum is 5 hertz. So we were measuring at 10 to be on the safe side and to have the data available for the eddy covariance approach, which I still haven't done, and I'm actually curious to see how it will compare to the bulk approach.
Yeah, that's very interesting. We are trying to use the same approach, you know, for fluxes of any dissolved compound, not yet on volatiles, but maybe soon.
Nice.
But yeah, that's the problem, not to have, you know, a continuous measure of the compounds at the same precision, no, at the same that the physical one, no. In our cases, we use oxygen sensors, and we want to, you know, calculate the fluxes for ammonia, for nitrate, for for everything. So yeah, that's okay. Yeah,
yes.
That's good. And also, I have another question. You mentioned that also corals are producing volatiles. Mm-hmm. So, in which magnitude? I mean, compared with symbiote, you mean without the symbiodinium interaction? No. Mm-hmm. So, do you know which is the mechanism they can produce? I can't imagine any cell type in the coral that can produce these compounds. I don't know.
So it's a paper by J.B. Rena, and it's I think either Science or Nature. The mechanism that they, which I'll be happy to send to you, they basically looked at they actually worked on recruits. So they looked at coral recruits that were aposymbiotic, and they measured the background level, and they measured the production of DMSP under stress in those aposymbiotic coral, and they saw an increase. I think that the mechanism behind is they think that there are some gene translocations because corals have been living in symbiosis with dinoflagellates for so long that it could be that there has been some translocations of genes occurring in the past that allows the coral to also produce the MSP. Well, the thing is, and that's probably one of the flaws of, well, I shouldn't say the flaws of the paper, but there has been some recent papers, I think in 22, showing that, or yeah, maybe 21, showing that bacteria can actually also produce the MSP. And the coral, we know, is an holobiont of more than the coral and the symbiosis. It also lives with a maze of bacteria. So it could be that in this experiment that they run, because there were bacteria there, they didn't work in sterile conditions. It could be that the bacteria played a role in the DMSP production. And that's something that, in fact, JB was worried about later on when he saw that recent studies have shown that the MSP can also be produced by the bacteria.
Cool.
So,
yeah.
Yes, but I'm not a microbiologist, but I believe it's like through gene translocations from the symbionts to the coral that the DMSP is produced. Yeah.
Good. A last question,
yes.
if I can. Yeah. Do you have any idea of any organism or any mechanism acting as a sink of volatiles in coral reef or what? I mean, question can be translated, because I always hear about producing, emitting, but nobody is using this compound.
I'm sure that bacteria play a huge...
macro things.
Macro things.
No, bacteria work. Yeah. Because it's I mean, yeah.
Yeah, that's true. I don't know.
Yeah, good. No, that's...
yes.
We will talk soon about
sure.
it. Thank you.
Related to the last but one question from Marta, in your experiment with juveniles, you said that you had just inoculated the juveniles within the symbionts. Is that right? So, did you do the same measurement before inoculation? No.
No, okay. We didn't, because that was our original plan. But the collaborators that we worked with, what happened is like the juveniles were unattended for a long time over the Christmas period, and so they were not going to be fed and so on, and we just decided that inoculation was probably best while they were inoculating their corals for their own experiment, that they will inoculate ours as well. But we would definitely love to go back and do some experiment on aposymbiotic juveniles, just to see what's happening, because then we will know before inoculation. That was one of the questions we wanted to answer. Now we are answering different ones, but one of the questions was like, yes, what portion of the BVOCs is produced by the coral holobiont, and what portion is produced by the symbiont itself, or therefore the coral alone, so with the bacteria. So yeah, we haven't done that, but we would love to, yeah.
Hi, thank you. What was the result, or do you know the results of the program of spraying clouds and shading, the large scale?
Yeah, so far what they just published a chapter on that, and which again I'm happy to share with you. They found that they could control they could modify the microphysics of the clouds, and they could increase the albedo, but it's not something that they can easily control and reproduce. So yeah. And also, of course, with this research, there is a big uncertainty around the upscaling, because it's one thing to have the technology to brighten one cloud as a proof of concept to see if we can master this technology or not. Yeah. But whether we can upscale it to the size of the Great Barrier Reef is beyond a concept at the moment. So it's and also making it sustainable in a way that right now we are using huge generators that probably emit more CO2 and methane than we are shading the reef. So, but it's the whole story behind was really a proof of concept to see if we had the technology to modify the microphysics of the clouds.
Thank you.
Yeah.
Any other question? I have one. So about this sad, but becoming more often bleaching events, right? And I remember that at Aslo in Mallorca, and many of us were there, Professor Hughes told us about this, and this is becoming more often and more. And he kind of, my impression was that he was not too pessimistic, meaning, okay, this is bad, this of course destroys part of the reef with the habitats and blah, blah. But what he says is that there is the kind of evidence for substitution or replacement of one species into other species that are more thermal tolerant and more resistant to. So we have to kind of get used to see the reef changing and evolving, maybe more than just disappearing, right? What do you think about this?
Well, I think that, yeah, for sure, as human beings, I think we want to preserve the environment that how we know it now, and that for sure, like evolution will lead. And well, I mean, I do think that the bleaching as we see it is definitely linked to anthropogenic climate change, and so that we are playing a huge role in these events becoming more severe, more frequent. But it's true that we're also seeing more resilience in a way, that I feel that often we will see like a huge portion of the Great Barrier Reef bleaching, and then meaning that it's not yet dead and it will recover quite fast. So that I don't know, and I don't want to say for sure that it's recovering faster than it used to. But definitely we are seeing some occurrence of new species and new clades of symbiodiniums, or new associations, I should say, that give more thermotolerance to the reef. So for sure, I think we will have new species appearing, some disappearing, probably some different combinations in between the symbionts and the coral that will happen over time. And well, so there is an effort as part of the reef restoration and adaptation program to artificially select the species that will populate likely the reef in the future. So they are doing not only selective breeding and selective inoculation, but the idea is to repopulate part of the reef that has disappeared with those species that we know are more thermotolerant, to again give a bit of a push to the reef. So yeah.
And no questions? My last one. And then about Heron Island. I mean, for those of you in the floor who are not familiar with coral research, Heron Island is a reference place. It's a beautiful island in the Great Barrier Reef where they do this type of research. And for me, it's been always a dream to go there someday. And I wanted to ask you, you've been there for, many times and for long. How does it work? Is it a research facility that anyone can use? It's run by a particular institution? What kind of facilities do you have there? You have to bring everything with you, or there are kind of equipped labs, or just an idea of how does it work?
It's well, so the island is divided in two. It's a really small island that you walk around in 45 minutes. Not if you really take your time. So it's small, and so you've got the research station on one side, a resort on the other side, and yes, you have to take everything with you, but there is a barge that operates every 2 weeks. So often what we do is we drive all our equipment, or you can ship them, obviously, if you're coming from far away, get everything to travel on the barge, and the barge comes every 2 weeks. So you plan your trip according to the barge trip. And and the research station is owned by UQ, so University of Queensland, but any researcher is welcome there, as long as you make your booking long enough in advance and so on. But yeah, they have amazing lab space, amazing lab facilities with flow-through water systems and aquaria and so on. So you can run, experiments over there, and you can access the labs and book them. Yeah. Yeah, no, it's they have great facilities.
And how do you access the natural environment?
From the how you access the reef? Yeah. Well, you're just there on the reef flat.
Yeah,
so are
there boats or?
Yeah, so you have boats available through the research stations. If you do research on the reef crest, most of the work that I've done, actually, I've never really dived on Heron Island for research, is that you can walk on the reef flat at low tides, collect your coral colonies within the scientific research zone. And if you were to do diving or more, and you need access to deeper water, you can just go with the boats of the research stations and book them, yes. So you don't go on your own, which is the case of One Tree Island. One Tree Island I've never been to, but it's meant to be even more remote than Heron Island, and you go with your little dinghy yourself, and you go and collect your coral and do your research. But on Heron Island, you go with the staff, and you can access different parts of the reef. Yeah.
Okay, actually, group, part of our group, we are going to Moorea Island in French Polynesia, which is another of these reference islands for coral reef stuff, and we'll be doing field work there. This is why I'm so much interested in how does it work in Heron Island. Thank you, Liz. Thank you for your talk. Thank you all for coming. And well, we will welcome Liz again in September or late August. So thank you for coming. Have a nice weekend.
Thank you.